All right, let's talk about lab. So this week in lab, we're going to determine the formula weight or formula mass of an unknown acid. So there's 3 steps to doing this. The first is to make your NaOH solution. The second is to standardize your NaOH using a known concentration of an acid. And this should be a standard acid, one that doesn't change over time, so that you can be really certain of the concentration of the acid. We may use HCl, which is not a good primary standard, but that's fine. And so then our NaOH will be the secondary standard. And then we're going to titrate an unknown acid using that NaOH and determine the formula mass. So, for an indicator, we're going to use phenolphthalein, which when it's acidic, it's clear, and when it goes towards a bit, when the pH increases, it's going to become pink, and it can get to be it can be anywhere from like a slight rose color to like very, very like magenta pink. So we'll see how that goes. Ideally, we'll have just a pale pink. Let's talk for just a second about the treatment of your unknown acid. So no touchy-touchy. You're going to be weighing by difference, which we're going to talk about in a minute. But when you're weighing by difference, it's really important that you use gloves to handle the vial of your unknown acid, because if you weigh it and then you touch it a bunch with your bare hands that may have hand cream or oil or whatever on it, and then you keep weighing it again, you might be changing the weight, especially because we're dealing with relatively small changes in the mass of the bottle. So let's explain that a little bit farther. So weighing by difference is basically you weigh the bottle with the acid in it that Amanda's gonna give you. Then you're gonna weigh it, record the weight, tap out a little bit of the acid into an Erlenmeyer flask, and then you're gonna weigh the bottle again. So the difference between the 2 masses is should be the amount of acid that you add to the Erlenmeyer flask for each treatment. You're going to perform these trials in at least triplicate, but you should definitely do your calculations in lab while you're just sitting right there, so that you know if you have good precision. And usually, if you have good precision, you're relatively accurate. You want to be sure that you make those calculations right then and there, right in lab, so that if it's not right, if you have one trial that doesn't look quite very good, then you can redo it. And so I think that when I did something similar to this, when I was taking this class, I did a ton of titrations, because I wasn't very good at them at first. So, because of that, because you have to do your calculations, or because it's most effective for you to do your calculations in lab, I want you to practice them outside of lab, and we can maybe go over them in class. But before lab, in addition to making your data tables, you need to do some calculations. So the first thing I want you to calculate is the concentration of NaOH. If you add 7 milliliters of 6 molar NaOH to 400 milliliters of solution. So just do a practice calculation so that you know roughly what concentration you're going to be dealing with. And you can calculate, once you do your experiment, it will be relatively easy to redo those calculations with your exact numbers. So then what I want you to do is calculate the volume of NaOH that you need to neutralize 30 milliliters of your 0.1 molar HCl. So that you know about how much you're going to be adding. If you have a 50 mil, you want to know if you're going to need 3 mils or 30 mils or 300 mils, so that you can figure out, so that you just sort of know what to expect when you're doing your titration. It's a very good idea. During my postdoc, I made like over 200 samples. And they weren't individual titrations, but part of the process was doing titration, so I did a ton. And the instructions that I read out of the book when I was planning my experiments, I read a procedure in a book to synthesize these minerals, and the recipe that was in this book was totally wrong. It suggested that you add like twice as much base as you needed to get this stuff to precipitate. And the first time I did it, you know, I had heard my postdoc advisor said, oh, I think there's something hinky with that procedure. So, you know, I went ahead and I was trying to be real cautious, and I overshot the endpoint, and it was a really specific endpoint, and it was really easy to overshoot. So I ended up actually doing a ton of those titrations, trying to get my experiments to be right. Anyways, regardless. So you want to know if you're going to have to add 30 mils, 300 mils, whatever. So do that calculation. All right. Then I want you to calculate the formula mass of I mean, you can use whatever numbers you want, but I want you to practice calculating the formula mass from this titration data. So say if you have 0.05 grams of an unknown acid, if you needed 30 mils of the NaOH that you are going to calculate the concentration of up here to reach the endpoint, what would be the formula mass? So do those calculations before lab, and it would be great if you did them before Monday morning so we could talk about them in class if necessary. All right, so lab write-ups. I want you to do individual reports with the same sections as we've been having. So purpose, introduction, results, discussion, conclusion. I want you to do these calculations by hand, and you're going to be doing them in lab. So easy peasy to just append them to the lab report. So that's all. Looking forward to seeing you guys in lab.